levels also have got really important contraindications, and many people with bacterial infections are old and comorbid. Even worse, we actually never find the pathogen in many patients, so we don't even know whether they really had a bacterial infection. So should we actually be comparing stuff versus nothing? But actually, again, a lot of these patients will die whether or not you find a bug. That's outside complicated UTI, which makes it very hard for a clinician to randomise against something which they feel might be a substandard SOC. And actually, it's even worse because for antibiotics, resistance means that actually we need diversity. One SOC will just drive resistance to that drug. But that means we need multiple trials against multiple non-existent different SOC, which many clinicians won't randomise to, many patients won't be eligible to receive for multiple reasons, leading to results which are simply not relevant to most of the patients, and results which accumulate very slowly and potentially have low power to exclude important differences. So, in a practical design, there is just no SOC. It's very simple. Each patient is randomised between a subset of regimens, and these subsets differ from patient to patient. In contrast, in the traditional platform designs that we've talked about this morning, there is still one SOC, which every patient has to have the potential to be randomised to. We do indeed test multiple intervention arms, we can start or stop. The SOC can change over time. You don't have to randomise to all test arms, but this primary analysis, as people have talked about throughout the day, is still a pairwise comparison with SOC in patients randomised to SOC versus each test arm. So going back to my example, here are my original 13 regimens, and there are just 8 example patients across the top, and these would be typical patients presenting with these serious CPE infections. And yes, no just means would you actually even consider one of these regimens in that type of patient? There is no regimen with yeses in every column. So instead, what would happen in a personal randomisation list is we consider for each patient what regimens might be relevant for them, and we randomise to those ones. It answers the clinical question, out of what I could use for this patient, what is going to give me the greatest chance of success? And that's because how the things that you could give this patient might compare to something else that this patient can't take is completely irrelevant, whether or not it's someone else's SOC. Now, of course, there's a trade-off, yeah? And that is really that you have to be able to argue that given the number of different options and the lack of SOC, choosing a treatment which is likely to be one of the best out of the multiple available options for that patient is more important than identifying the very best treatment, which is the type 1 error paradigm. What we really want to do is just avoid the worst. And so if you think about putting, an efficacy outcome like mortality on the x-axis and another outcome like resistance or cost on the y-axis, the SOC paradigm is identifying the very best treatment, assuming that the vast majority of patients can take this. Whereas the trade-off with the practical design is what we really want to do is work out what's the worst thing for each patient and avoid that. Meaning that if you have a new patient who can't actually take D, you still have information about how you should treat them. Implications, no frequentist testing, all the Bayesians say hurrah. Sample size is determined by information gained versus the current status quo. Becky will talk about that next. But I would argue that the current trials that we've got are really not designed to answer the question we need answered, which is what are the best regimens out of the available options for multi-drug resistant infections that will reduce morbidity, cost and mortality overall. And that's because there's no SOC, and practical is a design to answer that. Thank you.
Hi, everyone. So yes, I'm going to talk about the implications of using a practical design for how we analyse the data and how we determine sample size. And I'd like to acknowledge all the other co-authors who've been involved with this work. So as Sarah has explained, the motivation for using a practical design is settings where there is no standard of care. And in these settings, the focus is on ranking the treatments rather than testing. And this means that we have to do things differently, both when we're analysing the data and when we're determining sample size. For analysis, we need a method which is going to produce a ranking of all the treatments, and it must also take into account the method of randomisation, which is quite different from conventional trials. For determining sample size, there's no testing, so we can't use standard power-based methods. And instead, we've developed an approach which is about looking at how much information we would gain from running a trial. I'm going to continue with the same example that Sarah was using. So this is about carbapenem-resistant infections. This table shows 10 potential regimens for treating carbapenem-resistant infections and 3 examples of patients who would have different randomization lists. And as Sarah explained, in a practical design, patients would only be randomized between the treatments which are acceptable for them. So in analysis, we have to use statistical methods that respect that method of randomization. And direct evidence on treatment differences is only going to be obtained from patients who have the same randomization list. But we can also make use of indirect evidence by pooling evidence across randomization lists. So on this slide, I've shown on the right, I've shown 2 different ways of estimating the difference between treatments A and C. On the right, we have direct evidence being used. We're using evidence from randomization lists which included both treatments A and C. On the left, we're using indirect evidence. We're using evidence from randomization lists that included treatments A and B, together with evidence from randomization lists that included treatments B and C, and we're putting this together. Using indirect as well as direct evidence is analogous to what investigators do in a network meta-analysis, where data from multiple trials comparing different pairs of treatments get combined into a pooled analysis. Because the structure of the data is like a network meta-analysis, we can use a network meta-analysis approach to analyse the data from a practical design. So here we've got a subgroup of patients who have the same randomization list, and this is equivalent to a study in a network meta-analysis. And we're then pooling data from different randomization lists together in a combined analysis. If we do this, we're making an assumption that the direct and indirect evidence are in agreement. And this is true if the treatment differences are identical across all the different randomization lists. And this is an assumption we can test in our analysis. By using a network meta-analysis approach, we're going to get an overall ranking of all the treatments included in the trial, which is based on information from all the patients in the trial. We carried out a simulation study to evaluate our proposed method for analysis. We've evaluated the network meta-analysis approach and also approaches which pooled less evidence across randomization lists. We found that the network meta-analysis approach performed well under most scenarios. It gives us treatment rankings with good precision, and it's also robust to moderate variation in treatment differences across randomization lists. In practice, clinical decisions are going to be informed by rankings of treatments for both efficacy and safety, together with estimates of relevant treatment effects, and also other considerations like costs and availability of treatment. I'll now talk about how we can determine sample size for a practical design. So we've got a network of treatments and we're not doing any testing, so we can't use power-based methods. And instead, we're going to calculate how much value we get from a trial of a certain size. We suppose that we're going to use the trial's results to choose the top-ranked treatment for each patient amongst those treatments which are acceptable for that patient. So if our primary outcome is mortality, for example, we're going to look at how much mortality would be prevented by choosing top-ranked treatments based on a trial of a certain size, rather than making a treatment choice based on no trial information. And if there's equipoise between the acceptable treatments, then a choice based on no trial information is equivalent to making a random choice amongst those treatments. And this measure of mortality prevented is what we're using to replace power. Larger trials would provide us with more precise information and more accurate treatment rankings, and so they would provide us with greater mortality reductions. We simulate trials based on the planned trial design to work out the value of trials of different sizes. To do this, we've got to make some assumptions about how high we expect the treatment effects to be. So in the carbapenem-resistant infections example, I've supposed we're designing a trial to compare the 10 regimens I showed earlier. We are assuming that 30-day mortality is the primary outcome and that this varies from 10% to 30% across the 10 regimens. And I've then simulated trial data sets for a range of sample sizes. So this plot shows the average mortality reduction from based on trial information, in comparison with having no trial information. Because I'm stimulating the data, I know what the truly best treatments are for each patient. So I've plotted the mortality reduction we would get from a trial relative to the mortality reduction we would get if we had perfect information and we knew the best treatments. So you can see that the mortality reduction is increasing as the trial size increases. So if we have a sample of 250 patients, the trial is giving us limited information, and the mortality reduction would be 60% of the maximum possible reduction. On the other hand, if we've got a trial of 1,500 patients, then we're getting 90% of the maximum possible reduction in mortality. Based on that plot and some other results as well, in this example, it seems that a trial of 1,000 patients would seem a fairly good choice. If we were using top-ranked regimens from a trial of 1,000 patients, we would get 86% of the maximum possible reduction in mortality. We'd get a 70% chance of being within 2% mortality of the best regimen, and a 97% chance that the chosen regimen is better than a choice based on no trial information. And in practice, it's also important to vary the assumptions made about the design and look at the sensitivity of these measures. So in summary, to analyse a practical design, an approach similar to network meta-analysis works well. This provides us with an overall ranking of treatments and also estimates of the relative treatment effects. To determine sample size, we need to use a non-standard approach using simulation, and this allows us to estimate the benefit of trial results at various sample sizes. And finally, here are some references. Thank you.
Thank you very much, Becky. Again, any questions of clarification about that talk? Okay. So our next speaker is Mike Charland. Oh, go on.
Yes. Yes. So.
Oh.
Oh.
Do you want to?
Yes. Actually, can you repeat?
So even though some patients are only eligible or contraindicated for certain treatments and are only eligible for a subset of the treatments, you then provide a ranking across all of the treatments. How do you then apply that to the patient who is contraindicated to 3 or 4 of the treatments? Just choose the ones that aren't there.
Yes, that's right. So we provide an overall treatment ranking applicable to anyone, and then for each individual patient, there would be a subset of that ranking. So we would pick out... Yeah, that's right. Yes, that's right. Thank you.
your job as a clinician is to bring problems to statisticians and clinical trials units. And boy, did we pick a decent problem to talk today. So unlike the others, as a pediatrician, our trial is called NeoSEP, so you can probably work out what it's about. This is about NeoSEPs 1. So why is it needed? We'll talk through all of the bits and pieces. There is a huge problem of antimicrobial resistance in neonates. Neonates are a group of very vulnerable population globally, and as you can see here, actually the burden of antimicrobial resistance and of course the mortality associated with drug-resistant infections is a particular problem in the newborn as well. And there's high levels of resistance to the WHO antibiotic recommended therapy. In fact, the first line of antibiotics for is penicillin and gentamicin. It's been like that for decades and decades and decades and decades. In fact, since the very early use of antibiotics in neonates was a long time ago. There are hundreds of thousands of babies unfortunately die per year due to multi-drug resistant pathogens. And as Sarah was talking about, sort of these ESBL type pathogens dominate as the sort of leading cause of that. And we've recently had the Gram study, which has sort of emphasized the burden of that. So I'm going to talk briefly about the recently through GARD-P, the Global Antibiotic Guarantee Partnership, and Gates sponsored a global neonatal observational study, which finished off in about 20 hospitals in a dozen countries. And that was 3,200 infants that designed to inform the NeoSEP1 trial, and the analysis of this was done by Wolfgang and Sarah. So, the main findings is there is a huge variation in antibiotic use. So, though Sarah says there is no SOC, actually, well, we have over 200 empiric antibiotic regimens being used to treat these 3,000 babies. So there was, there are around about actually something about 250 antibiotics being used globally, and nearly any combination of any antibiotic you might possibly ever have heard of are being used to treat the babies. You will note the lovely colour scheme, which of course is the new AWARE classification, acts as what's reserved for antibiotics, had to put that in. And you can see that first line antibiotics, pen and gen, second line antibiotics, more yellow, sort of third line antibiotics go more dark and then sort of red. So there's a huge variation of standard care, and a very high use of second line antibiotics as first line treatment. In many settings, there is so much resistance that actually a rapid escalation to the sort of last resort antibiotics, meropenem, is very quick indeed. So there's also switching. So obviously babies, it's very difficult. You're not very well as a baby. You don't do much. You just don't lie there, and you just don't lie there even less. By just sort of kind of, and so that's quite subtle, and therefore it's difficult to tell really what's going on. And therefore, if they're not getting better very quickly and sort of waving and crying, then what you do is escalate to the next regimen, and you keep escalating. So there's a very high rate of switching of antibiotics on, and often that's not very much in the way of stopping antibiotics. Much more commonly, that people would just escalate rapidly up as well. And in comparison to the usual guidelines as well, of the antibiotics guidelines, actually there's not much use of those antibiotics. People have just given up often in the early ampsies, and of course they're now last resort. Sort of colistin antibiotic is now increasingly being used in many units globally, particularly in sort of Southeast Asia and Asia. So we know that it's very challenging. We know that there's lots of bugs. We know there are very few neonatal antibiotic trials. When we say very few neonatal antibiotic trials, you've probably looked at ClinTrials.gov recently and put in the words COVID, and there's over 5,000 studies.